Okay, why don't we get started? I'll just say very briefly, welcome everyone to Symposium 5, and we'll start with Dr. Shiang Hu. Please go ahead and begin, and we look forward to you sharing this session.
Okay, thank you, Christine, and welcome everyone to participate our symposium. Our symposium, the topic is EEG modeling. Mainly, we will have 4 speakers to introduce their work progress on the EEG spectral and source localization and brain stimulation. And the speakers we have is myself as the first one, and Dr. Ying Wang, and the third one, Dr. Ronaldo, and the last one, Miss Anisleidy. Okay, I'm the first one. Now I'm going to share my screen. My topic is about a neural power spectral decomposition model. And we firstly describe a nonparametric model to split the neural power spectrum. Actually, let me show split. The work we generally did is to split the power spectrum no matter in which scale you estimated in the scope or in the source, and we can decompose in the background spectrum and the oscillatory spectrum. Okay, first let me give some introduction. Briefly, very briefly, first one, this is the biophysical modeling of the generation of EEG current, we obtained the temporal, the traces, the waves, but we don't study in the temporal domain. We in the quantitative EEG field, most of the studies looks to pay attention or looks for the frequency characteristics. And since the discovery of EEG over the decades, people generally pay attention to the narrow bound frequency ranges, such as delta, theta, alpha, beta, and gamma bound. And each of them are well corresponded to cognitive function and mental disorder. But how do we precisely study these oscillatory patterns, and how do we parameterize it? And recently, people are working on the neural oscillation parameterization, and we firstly need to clarify the definitions of neural oscillation. The neural oscillation doesn't generally refer to the EEG traces. It is actually the rhythmic or the repetitive electrical activity may be generated in the resting state condition or in the invoked by the external stimuli. And how we study the neural oscillation, this is the spectral wave, and we apply the transformation with basis function, use sine and cosine. We can plot the power spectral density with this, and more generally here as a special case here, we partition the general frequency bound into different narrow bound. And the thing is, there is a general monotonously decreasing trend from the low frequency to high frequency. And also in the low frequency, there actually is a large proportion of all the power. And how do we study the power? We need to show a spectral peak. And all the spectral peaks, this prominent one, this less prominent one, also the trivial ones, they are reading over the background spectrum. And actually, the red spectra, the red line in the last page, the background spectra is originated from the arithmetic electrical activity, or called aperiodic activity, which is maybe generated also in either resting state condition or in response to stimuli. But the mechanisms, neurophysiology mechanisms are not clear in this aperiodic activity. There is mainly two hypotheses before the synaptic timescale hypothesis, which may be formed by the Lorentzian process. And the second one is the self-organized criticality avalanche dynamics. And this year, just this spring, there's another paper reported a neurophysiology model for the 1/f aperiodic activity using subcritical network dynamics can generally well model that process using a branch parameter as 0.98, and which is publications in neurocommunications. And I just show their result here for the detail, and we can check this paper later. And how do we really study the neural oscillations? In 2013, it's Lopes da Silva published in the Neuron a paper to say that we have to, if there is a well-defined oscillation exists, we have to identify and show that there is a spectral peak and its peak frequency, bandwidth, and power. And over the decades, there are multiple attempts to address this issue. And dates back to 1970, there is a hardware filter design to obtain the alpha rhythm. And the Peterson, which is nonlinear modeling, did a lot of this kind of work, and to split this by alpha, the multiple overlapping alpha. And in the 1989, Robert Pascual is trying to decompose the alpha and the background spectra using student T function here, but which only pay attention to the alpha spectrum, alpha peak. And in most recent, in 2018, the Heller from Brady Wojtek group wrote a Python package and called FOF, and they can quite conveniently and quickly to decompose the spectra, but in the logarithm scale, the different peaks with Gaussian curves, Gaussian kernels, and the 1/f power law functions to get the parameters. But is this really right? Correct. The FOF model actually is a parametric model which simultaneously estimates the parameters during the fitting process, which means in the fitting process, it will fit different process in the log scale by log transform the spectra into different components, and at the same time will return the parameters. For example, the power law offset and exponent. And how do they check for each peak? It use thresholding effect and iterative least square. And the point of our work is we think their model is firstly maybe limited to the hard kernel functions in fitting, which is inflexible. for example, the Gaussian condition and the power law condition. If we check the intracranial EEG spectra, we from this, for example, from the precentral gyrus, we do the clustering. We here 3 plots, and you will see here the spectral shape may be quite irregular, and the peak is not is a bell curve or is concave upward, concave downward. It's maybe show some ripples or is quite regular. So so we worry if FOF can really output enough result. So the limits of FOF may be not that flexible, low flexibility, and also is because its decomposition in the log scale is actually equivalent to multiplicative mixing. So we are trying to give more natural model following the natural shape characteristics and its mixing phenomenon, non-parametric model. Okay. Then we assume the Fourier series mixing model, which is unobserved in the latent space, and all the components can actually the spectra is the second order variance of the Fourier series. So we apply the variance components model, we can obtain this formula. And observed spectra is by a periodium or the multi-taper or the other spectral estimation method. So actually the work now, the problem is transformed given PF, the real spectra, observed spectra, and the number of peaks. And with respect to the unobserved, the latent variable to estimate the spectra of different process. This is solved by the expectation maximization algorithm. And theoretically, Y2 likelihood, especially the non-parametric Y2 likelihood, is the statistically consistent estimator to the spectrum. And we applied this as the criteria to decompose the different process of the spectra. And we need to solve the problem for EM as the starting point is to define the initial number of peaks. We solve this by checking the peak prominence, and which is you may use the built-in function in MATLAB or Python or available in other packages called find peaks. And the natural shapes characteristics is shape constraints for the background spectra. We only apply the monotonicity decreasing, and also for the one peak we cannot guarantee. We cannot strongly make the hypothesis that is a Gaussian curve. We only think it's a unimodal or monotonically firstly increasing and then decreasing. And this is not a spectral peak. This isn't either. So the basic the flow chart is then we'll after we input the spectra, we define the number of peaks, and we initial fit it using student T function, have a rough fitting, and then we submit it to the EL algorithm for iterative for multiple iterations, and based on the Y2 likelihood criteria, and finally it will converge and will return different components. And our model has the feature is individual fit consider the monotonicity. Is a model to decompose in the linear scale without log algorithm transformation. And theoretically, it allows for unlimited number of peaks to decompose. And our result is validated from the simulation by special cases and batch simulation for quantitative error analysis and special cases for qualitative error analysis. And for the special cases, we check for the overlapping and skewed and concave upward very that are typical shapes. If we look at the source spectra or intracranial EEG spectra, SEEG spectra. So it shows our model, parametric model is poor and in fitting irregular shapes. Multiplicative may enlarge the fitting error in the log scale. And non-parametric model is more flexible, okay? In the batch simulation, and we get the results, we simulate over low frequency, high frequency by varying the number of peaks. And the CPI has much lower error than FOF, and FOF may much easier to overestimate the peak number. CPI may ignore the tiny peaks at the higher frequencies. And we further validated our model using the real data, sleep staging and sleep staging task, and apply the CCSHS sleep EEG, and we apply the CPI and FOF. You can see here, after with the CPI, the included angles between different solid curves are larger than FOF, which means the CPI model helps identify the more differentiable features from the aperiodical spectra than FOF, and the result is indeed like that. And then we check the oscillatory peaks discovery, and we use more than 1700 of intracranial EEG, which generally covers the whole cortical space and from the healthy brain regions over 106 subjects. And this standard is reported by the clinicians, and they are neurosurgery clinicians. They checked by visual inspection by quite handcrafted work. And we compared our model and with FOF. We found the CPI is much closer to the standard result. So as a summary, nowadays spectral decomposition is a hot frontier, and nearly all these models are the adapted variants of the FOF, and only CPI is we think only CPI is quite distinct from others because it only considers the natural shape characteristics and using the statistically consistent estimator of the spectra, and without logging transformation spectra is the only a true model by linear in the linear natural scale to decompose the model. Its advantages are robustness to regular shape, flexible, allows for flexible parameterization as the user preferred, and it can be widely applied in the neurofeedback and normative modeling and process auto-diagnosis. And at the end, I want to say that CPI is suitable for quantify EEG, SEEG spectrum oscillations, and can also serve an alternative of FOF to study the scope EEG oscillations. Also, our paper is finally selected by the editor as the featured article in the IEEE GBC journal. Our code is all published available in the GitHub with walkthroughs and the introductory guide. Okay, thanks for all my collaborators like Dr. Pedro Sosa, Professor Yao, Dr. Jorge Bosch, and helpful discussions with Dr. Ludovico, and thanks for your attention.
Hello, everyone. Here I will continue the topic on the spectral domain and parameterization. Here I will first address my result here. The sleep process you saw in the last presentation is linear and Gaussian, and the alpha process is nonlinear and Gaussian. Here, I will prove this with the model called the high order C alpha model. And okay, you know that we have released a multinational QEEG development service, which is fitted by a suite of non-parametric functions. However, this non-parametric function is very hard to do the statistics, and it's hard to get the future of the biophysical means. So the parametric method is very useful here. Here is the two examples that parameterize the spectrum of this surface, the cut of this surface, like the C alpha model and the FOV model. And this is the list of the study of parameterize the spectrum, specifically in the EEG spectrum. And here is the history of the studies done by our lab. First is the paper from Pasqua. He did the CF model in 1988, and then is the paper from Professor Pedro. He used the PCA to find the C processes, the short range, like the spherical harmonic noise process, and the alpha process, which has a long range coherence in the brain. And later on, the Dr. Hu has represented his C pi model, which he initiated published this in the archive in 2019, and later Pasqua improved his method to get the surface-based the source of the CF model, which use the non-negative matrix factorization to get the C and alpha process in the source space. However, this previous methods are all based on the second order statistic of the process. These are sufficient for the linear and Gaussian, but Turing is highly nonlinear. So we need a model that represents the non-linearity and non-Gaussianity of the brain. Here we use the data from Montreal Neurological Institute, which has 1,700 electrodes from 106 subjects. In the original study, they already discussed the peaks in different bands. And okay, here we use the bispectrum, also the normalized bispectrum to represent the nonlinearity and non-Gaussianity. And this is a harmonic-based model. And why this harmonic-based model is working? Here is some example. First is Robinson and his group use the resting state data and fit with several Gaussian peaks, found that the harmonic relationship between the bands. And then some tasks, especially the steady state visual evoker potential, that when we input the sine wave to the vision, we found the harmonic, different order of harmonic, even the subharmonic, and the entrainment of the EEG. And they model this with a neural field and the neural mass, and reproduces the similar phenomenon. Also in movement and discrimination task, they found the harmonic phenomena also. And why use this high order spectrum can represent this harmonic relationship. Here's a example, a simulation example that we simulate the Stuart-Landau model. First, in the top left is the Stuart-Landau with the pure Gaussian noise, Gaussian innovation. And you can see the spectra is only one peak, and there is no value highlighted in the bicoherence. And in the top right, this is the Stuart-Landau with a non-Gaussian input. You can see there is a constant bias on the bicoherence, and the spectra is similar. And when we add the quadratic term to this Stuart-Landau model, we can find the symmetric round circle change it to a triangular-like circle, and the harmonic is appear, and you can see the bicoherence can represent the relationship of this harmonic. And like Victor just said, this is a symmetric breaking of the network. And when we add the non-Gaussian, you can also see the constant deviation of the bicoherence compared with the Gaussian innovation. Okay, then based on this metric, we can perform a test of linear and Gaussian. First, we fit a linear autoregressive to the time series and get the residue. With this residue, we fitted a density function to it, and with this autoregressive and this density function, we get 3 types of bootstrap samples. First is the linear one. The linear is the linear autoregressive at the innovation resampled from this the residual density function, and then is the symmetric version of this residual density function to generate this called linear symmetric process. And the final is the most simple one is the Gaussian process. We use the linear autoregressive model fitted to and add the Gaussian innovation. From the diagram of spectra, you can see all these 4 looks the same, but the bicoherence is totally different. The original data is it has nonlinearity, and you can see the highlighted dots there, but the bootstrap sample, there is no value in the bicoherence. With this test, so you can see that even the spectra is the same, the system can be different. So we need the bispectral for modeling this. So with this test, we tested the whole data set. We found that more than 70% channels are nonlinear or non-Gaussian. And the distribution of this the p-value, you can see the most nonlinear non-Gaussian channels are located in the occipital and the parietal lobes. Okay, then is how we build the model. First, we need to know how these components organized theoretically. First, if the components are 2 independent process, then we can directly add them. Since the independent process, each order of the cumulants can be adds up, and Fourier transform is a linear operation. So the cumulative spectrum also can be adds up. Then is the C then is another case if the C process is the innovation of the nonlinear system, which means the C process is the input of the alpha process. Then we can prove that so the components we observed is a multiplication of two processes in the nature scale, which means which is is additive in the log scale. This can prove that with this Fatera expansion. Also, with this expansion, you can know the relationship between the peaks are sum or difference of the fundamental frequency. And the peak of on the bispectrum, it has a relationship with the spectrum. This all is all caused by you can use the recursive you can refer the here is example of the quadratic nonlinear system with Gaussian innovation. You can see all the output spectra is the function of the first order transfer function, and the bispectral is also additive according to this formula. Here is other reference that you can find with the system with a non-Gaussian innovation, and even more general, you can build the model with a generalized frequency response function. It's more complicated, and up to now we don't have enough information which of the choice is correct. We only know the process is generated from different layer of the cortical through the laminar study. The C process and the high gamma process is generated from the shallow level of the layer, like the first and second, and the upper process is more deeper in the fourth or the fifth of the layer. Here we use the simplest assumption that the two processes are independent. So our model is the additive model in the nature scale. Actually, when you perform the fitting multiplication or the additive is the result is very similar. And with this model, we also we also get a likelihood function of this model under the asymptotic asymptotic Gaussian property of the log spectrum and bispectrum. And here is the detail of the components of the model. And actually, our T function, the Cauchy kernel or the Rollins kernel is for the transfer function. Here you can see the example for intracranial data. You can see the background trend and the alpha peak and beta peak. We call here the harmonic of the alpha. And you can see the relationship between the peaks, like some dots here. Here is another example. You can see the which is generated by a phase-coupled harmonic oscillator simulated with a non-Gaussian noise, and you can see the background noise and the fundamental oscillator and oscillation and the harmonics and the subharmonic here, and this is the third order spectrum, the normalized third order spectrum, the bicoherence here. So this is the magnitude of the bicoherence. Here is the real part, and this is the imaginary part. You can see the empirical one and the model one is very similar. And with this model, we can separate the C process and the upper process in the bispectrum. To get here is the bicoherence we define here of each process. You can see the C process is like the non-Gaussian, which is a constant deviation of the whole plane. Why this have some lines here? Because the spectrum used here is the whole the total spectrum to get the normalized. So we have a small value here, and here is the alpha the backcoherence of the alpha process, which is very sparse dots, and only appear where the harmonic have a relationship. Here is the result for some channel which is not which is imperfect harmonic phenomena, but you can get why this model is better than on spectral based only model. A spectral only based model cannot find something. Like here, you can see the first you see this this is the empirical bispectrum bicoherence, and this is the fitted bicoherence. You can see this is very high value here, which means this fundamental oscillation is coupled with this frequency. But if you only fit with spectrum, you cannot find the peak here, and you also cannot know they are phase coupled in the signal. And here is another example that you can see two larger peaks there, and some other harmonic there. But in the bicoherence, you can see the two large peak has not that does not phase coupled.
Sorry, Dr. Wang, we've got about 2 minutes.
Okay. Then I will present the result of the group level test. Here is the follow of the test for each channel. Let's see the table. Here is the first. Here is the sum of the bicoherence. We with the robust mean we test the group. C process is accepted, and the alpha is also accepted. For the individual channel, most of the channel are accepted, which means the C and alpha is because the sum of bicoherence tests the Gaussianality, so which means both are Gaussian process, and we also test the maximum of the bicoherence. Follow the Hinich test, and the C process and the alpha process. Oh no, the C process is accepted, and the alpha process is rejected. You can see the rejected channel is very close to the global test in the previous slides. So here I will present the conclusion. The C process is linear and Gaussian. Alpha process is nonlinear Gaussian. Most peak are the harmonics of the alpha. C process is the noise or innovation of the system. C process is not generated by the chaotic system because we don't see the value in the bicoherence of the in the empirical data, and the model can describe the energy propagation between the frequency. Okay, that's all. Thank you.
Okay, so my name is Ronaldo. I'm a member of the Cuban Neuroscience Center and also from the Young Cuban Channel Laboratory and the Global Brain Consortium. I'm going to speak today about the Chi-alpha model net. It's a model for mapping effective connectivity, especially across extensive EEG and MEC datasets. So we speak a lot of these days of gathering data, but we believe in our lab that at the same time, in parallel, it's necessary to develop methods to analyze this data that we are going to gather. We also speak about an effort of the Global Brain Consortium and our lab in the harmonic EEG paper, where we was able to gather data from 2,000 different patients across 9 different countries and multiple devices. We can see here in the right the histogram of the population distribution, again the age, and basically what we was able to prove in the harmonic EEG paper is that from the cross spectra in the scalp, you can elaborate clinical norms that allows you to classify different type of pathological subjects. But as we know from the literature, say Pedro, Freeton, even Babiloni that is in here, the analysis of the cross spectra in the skull is a bit about different kinds of artifacts. So that you can mention in here bowing conduction effects, spurious connection, this is a fake type of connection. So we need to move to source analysis and we need to perform effective connectivity models in the source space to reduce these kind of artifacts. So what are the kind of effective connectivity models that is known in the literature? This is the important ones for our discussion. There are many other types. And you have the ones that enable sequential estimation at each frequency. And basically here, what you do is you take from the EEG, you map the EEG at a specific frequency to the activation at that frequency, and from that activation to the core spectra at that activation. You have also parallel estimation at each frequency, and what you do is basically on each frequency, you map the core spectra in the skull to the activation in the source at that frequency, and in parallel also the core spectra at that frequency. And the problem that these two types of effective connectivity models have is that the size of the data that you are producing for every subject in the source space increase with the number of volts into the square. So this becomes a problem if you want to perform high resolution source analysis in large data set. I'm speaking here of 2,000 subjects or with a space in the sort of 8,000 voxels, something like that, or even higher data set with 10,000 subjects. That is what we want to do. So we are proposing here to try to solve these problems by proposing a new type of model that enables joint estimation across frequencies thanks to anatomical and spectral constraint. We call this model Shiang-Hu model. The name is going to be clear in our presentation. And basically what this type of new models do is that you take the full core spectra in the skull and you map it to a set of parameters in the source space. We can see here in the pictures. And this set of parameters doesn't depend on the frequency and the time depends on and the time. And this set of parameters also parameterize you the full core spectrum in the source. The really interesting thing in here is that the size of data that you produce for a subject in the source only depends linearly with the number of voxels. So this is particularly attractive for the study of large data sets. That is what we want to do in the future, move from 2,000 to 10,000 and even future. So the Shiang-Hu net has different type of constraints, and the first type of constraint is anatomical constraints. The anatomical constraint that we are going to use in this paper is the anatomical connectivity, the length of the tracks, and this data, we get this data from the Human Quantum Project. Here we have the paper. And we also are going to use the delays, the delays atlas of the conduction delays between neurotracks. And this data, we can find this data thanks to the FTRAT Consortium in the recent paper that they estimate the mean delay atlas from 360 patients, but they give only a mean value. They don't give a lifespan of how this atlas evolved with the gain. That is really important to these normative studies that we want to conduct. So basically what we do in our paper is use all this anatomical information that you will have for the Human Quantum Project to fill this missing data, to fill all this missing data in the delay matrix and also find the lifespan of this delay atlas. In the Shiang-Hu net model, we also have spectral constraints. We have a long history in our lab and also in CNeuro to working in spectral constraints. You have here the work of Bosch, the amazing works of Pascual, also the works of Pedro, and also the work of people that are outside of our lab. We can mention in here Boyted in his famous paper in Nature. And what are spectral constraints? Spectral constraints are basically regularities that you can find in the frequency domain, but also in the spatial distribution of all these processes, of these spectral processes in the brain. So what is the SHI process? The SHI process is like a type of decay with the frequency. We call this 1/f decay. We call this decay SHI process. And we can also observe like a type of peak in the alpha frequency band, and we call this peak alpha frequency. And when we know also about this regularity in frequency domain, we know that, for example, that the gain causes like a shift from lower to higher frequencies of the alpha peak in the alpha band. We can see here in the picture that the frequency shift from 5 years to 14 years. We mentioned also that we have some type of spatial regularities of this type of process. We can see in this paper published in Nature of Wojtek in 2020, he was able to establish for a study only in 18 subjects that the alpha process takes place primarily in the occipital area of the brain. You have also some activity here in the parietal lobe. And the aperiodic component of the spectra, that is basically the shift process, encompasses the whole brain. So we want to try to replicate and display this kind of data and use this constraint. So I'm not going to present all the mathematical details of the model. You are going to have that in a bioarchive and a publication in the future. I'm going to explain here the Bayesian configuration of the Shiang-Hu model and how we integrate all these type of spectral and anatomical constraints in the design of our model. So first, basically, you have the classical observation equation that connects you the observed EEG with the activations in the source. And the first assumption, an important assumption they are going to make, is that this activation in the source can be decomposed into an activation due to the shift process and into an activation due to the alpha process. If you have additional processes, say theta, beta, you can suppose that you can decompose in the sum of all these processes. And then you are going to assume that all these activations associated with these spectral processes, the G and the alpha, follows an autoregressive model where the spatial temporal correlation of these autoregressive models are determined by the anatomical information that you have, saying here the anatomical connectivity, the length of the tracks, and also the delay matrix. And where you integrate the G and the alpha in this model. The G and the alpha is integrated in this model, like in the innovation in the autoregressive model. And finally, what we can observe in here is that the parameters that for our model, the parameters that describe the scroll spectra in the source for our model are time and frequency in the plane. So this is particularly interesting for analyzing large data set. You also have a set of hyperparameters to your model that will promote sparse models. It's also important to remark that in our model, the delay is also a hyperparameter. A delay between two voxels in the brain, between two regions in the brain, is a hyperparameter in the model. So the first result is that we can use the Shiang-Hu model. This is the first result. What we can do with this model is that we can use the Shiang-Hu model to map delay atlas at a specific age. So how you do this? So basically, you have the cross spectra of 2,000 subjects. We want to have data for 10,000 subjects. This data span across different age groups and also multiple countries. And when you are going to use this data to find the likelihood of the hyperparameters and also the parameters of the Shiang-Hu model, and then optimizing the local likelihood using Markov-Chain Monte Carlo, you can find which delay between voxels in a specific age are the most probable one given the data. And we are going to use, really important here, we are going to use for this Markov-Chain Monte Carlo, we are going to use all the anatomical information that we have from the FDRI Consortium and the Human Consortium project to feed like a solution space for the hyperparameters that we are going to find. So what we get using this, what we get is a map of delays between voxels and also for different ages. And if you feed the data, the delays versus age, and also the length of the track on the Human Consortium project, you get this nice surfacing here in the right in the top, where you can observe the vertical axis delays in milliseconds. You have the length of the track in millimeters, and also the years is time. And what you can observe is that the delays of conduction, the conduction delay between regions in the brain that our model is estimating based on these 2,000 subjects for every age, the delays tends to increase with the age, and we have like some type of decrease in here on between 45 and 15 years, and you also have that like this tends to increase of the length of the tracks. Of the delay with the length of the tracks. And here we can see a regression of a partial delay lifespans of the HSP atlas with time. So this is really important for two reasons. The result we have here is important for two reasons. The first one is that you can use a Chi-alpha net model not only to fit the cross spectra, but you can also use these models, these effective connectivity models, to obtain a lifespan of delays at specific age groups. And the second reason why this is important, suppose that tomorrow Victor Yesa want to make a detailed neural field simulation of the brain that have 16 years. For that, he needs a spatial distribution of delays in the brain between neurotracks. And what we are proposing here is which type of delay can be used in there, based on a data set of 2,000 pages. So it's a strong result. And now we are going to prove in here in this slide is that the shift process is a process that encompasses the whole brain, while the alpha process is a process that is primarily focuses in the occipital area of the brain. The first two set of rows in the picture in the left, you have here the estimated lifespan of the population probability based on 2,000 subjects of find a particular spectral process in one particular voxel in the brain. And what we can see is a trend like the alpha process to be ubicated in the occipital area of the brain, while the shift process is more uniform in the brain. And in the second set of rows, what you can find in there is for which one of the voxels, the probability of a particular voxel, a particular process in a voxel is higher than 90%. So what we can see in the right is a simulation of a regression of the Chi and the alpha process against age. We can see fluctuation, but the result is the same. So the next slide, going to be the last one. What we can see here is a population probability distribution of the parameter of the Chi-alpha model that we are estimating here. And the main important things in here is the developmental trajectory of the alpha peak frequency. If you remember, we mentioned it before when we speak about the spectral constraints, that this is like a kind of shift of the alpha frequency with the age. And what we can see in the regression in here, the blue point is the initial point of the alpha frequency at 5 years. And we can see that again produce like a shift from lower frequency to high frequency in the alpha peak band. This is estimating from 2,000 pages, and we want to move that to higher.
Thank you very much.
This is all, and thanks you all guys. This is my contact if you want to get involved with us to get more data and make this even stronger. If you have any question, feel free to ask.
Hello everyone, my name is Anisleidy González Hans, and it's a pleasure to be part of the GBC. Today I'm gonna give a brief talk about improving brain simulation, accuracy, sensitivity analysis, and realistic time delays in neural mass models. This study is a collaborative effort that was originating during my PhD research at Pedro's lab in USDC and is currently ongoing at Alan's lab in the Montreal Neurological Institute in Canada. This study addresses a critical gap in current neural modeling, primarily tied to the classical Janssen and Reed neural mass model. The lack of a general theoretical formulation hinders modularization and the treatment of delays, particularly distributed delay, which have not been explicitly modeled until now. Recognizing the need for improvement, we focus on algorithmic efficiency in calculating prior inputs for each neural math. This presentation aims to underscore the urgency of refining our approach to neural modeling, emphasizing both theoretical advancement and practical applications. Our central hypothesis revolves around enhancing the efficiency of neural mass models simulation through two keys of methodology, the local linearization method and the strategic utilization of the connectome tensors. The primary objective is to design and develop an efficiency algorithms for the analysis and processing of high-dimensional data. The algorithm will be specifically grounded in the utilization of connectome tensors, aiming to enhance the overall capabilities of data analysis and processing in a high-dimensional context. The overarching aims of these projects are seamlessly incorporate connectome tensors into neural mass modeling with a particular focus on addressing challenges associated with distributed delays to enhance precision, critically examine current integration methods in neural mass models, identifying and illustrating limitations and challenges, paving the way for informed improvement. Simulate and demonstrate the impact of distributed delays and various connectivity topology within neural mass models, providing a comprehensive understanding of their influence on model behavior. Extend the Janssen and Reed neural mass model to incorporate a broader perspective with 1,000 cortical columns to enrich the understanding of neural dynamics at a larger network level. Study the influence of neural mass models parameters through a sensitivity analysis of the model, design and implement an efficient algorithm along with the creation of a toolbox facilitating the seamless integration of high-dimensional neural mass models and offering a valuable resource for neurocomputational endeavors. Neural modeling serves as a valuable tool for bridging the gap between experimental observations and theoretical predictions, providing a means to integrate empirical data with mathematical frameworks. These models are typically described by differential or integral equations, and a key aspect of neural modeling is the division based on the level of detail being modeled. At the microscopic level, models focus on the behavior of individual neurons and their intricate interactions. In the microscopic level, there is a huge amount of equation, and an example of that is the Blue Brain project. And on the other hand, at the mesoscopic level, neural models take a more macroscopic approach by considering population of collective behavior of neurons. At the mesoscopic level, a more manageable amount of equations are defined, and a mean field approximation is an example of that. And we have, for example, the DCM, the Brain Dynamic Toolbox, and the Virtual Brain. Mesoscopic levels are divided into neural field models and neural mass models, and our work is concerned with improving neural mass models. The Janssen and Reed cortical column model, also known as the Janssen and Reed neural mass model, is a mathematical model that describes the dynamics of a cortical column at the mesoscopic level. It aims to capture the essential characteristics of neuronal activity within a cortical column, particularly in relation to the generation of alpha rhythm observed in the EEG. The model consists of 3 populations of neurons that represent different brain regions and is based on the interaction between excitatory and inhibitory interneurons. Some researchers such as Wilson and Cowan, Freeman, and Lopez da Silva have made significant contributions to neural mass model by using stochastic differential equations in their work. By incorporating stochasticity into the model framework, they capture the variability observed in neuronal activity. Among the various neural mass models, the Janssen and Reed model stands out as one of the most well-known and widely used examples of this approach, and in this work we also follow this formulation. Neurons are the fundamental building blocks of the brain, playing a crucial role in transmitting electrical and chemical signals. They serve as information carriers, facilitating communication between different regions of the brain. However, it's important to note that this transfer of information is not instantaneous and is subject to delays. The speed of signal transmission through neuronal connections can vary due to various factors, including the length of the connection, the conduction velocity of the signal that is affected by different axonal properties such as the length, diameter, myelination, and G-ratio. These delays in information transfer have been a subject of interest in neuroscience, and researchers have developed models to capture and understand them. In some models, these delays are considered as instantaneous axonal propagation, assuming that the transmission occurs without any time delay, while others interpret these delays as a Dirac delta function. New studies in the field have shown that delays between different parts of the brain cannot be described by a single point in time, and researchers like Innocenti have discovered that the speed at which signals travel in the brain can vary because of factors like the size of the pathways. This means that delays should be seen as a range of possible times rather than just one specific moment. There are some researchers, such as Mark Dragsmith, that have been really involved in computing and improving ways of calculating conduction velocity. And as we can see here, the mapping of conduction velocity to axonal diameters appear approximately linear, while the mapping to G ratio follows an inverse log square root function. This approach has been shown to give a valid aggregate measure of G ratio for a distribution of axonal diameter. In this case, P is some constant of proportionality, which was estimated in their data to be 16.99. Conduction velocity was most sensitive to a unit change in G ratio by a considerable margin, and axonal diameter has the second highest sensitivity, as we can see in this table. In order to compute the conduction velocity, we need to firstly calculate the axonal diameter, and for that we follow the Sepperband work, who focus on finding an accurate distribution to represent axonal diameter. He explored with a long list of probability distributions, and he found that the generalized extreme value function was the better fitter for axonal diameter composition. What we propose is to take advantage of their finding, and for that we look into Caminiti and Leward work based on diffusion tractography and electron microscopy of the corpus callosum, and take from them the mean and the standard deviation that we need for computing the axonal diameters. Having this distribution, we are able then to compute the conduction velocity. Therefore, in order to calculate the time delays of signal propagation, only the length of fiber tracks is needed, and we extracted it from histological data analysis made by Caminiti in 2013. I followed the Janssen and Reed neuromass model formulation, and the new approach in this model is the definition of the connectivity tensor, including temporal dimension with arbitrary delay instead of using a connectivity matrix. This concept is crucial because connectivity involves more than just indicating which neural masses are connected and the strength of their connection. It also involves the distribution of delays in information transfer along each connection. I implemented a brand new type of realistic conduction delay. For that, 3 types of delay tensors were explored, given by instantaneous, synchronized, and distributed delay tensors. I want to highlight that other delay tensors models are possible and easily to incorporate into this framework. The other component of the connectome tensor is the connection strength, and for that I define 3 types of connectivity topology. The nearest neighbor, where only short connections are included. The small world network, that keeps the short-range connections to neighbors and adds random sparse large-range connectivity. And the full connected network, that includes all the connections of the graph. For numerical integration, we use the local linearization method to embrace local linear approximation instead of global linearization. This method preserves the original system dynamics and unique feature compared to traditional method. This strategy guarantees non-explosive behavior in discrete time system, providing stability to the model. Achieving this stability involves stochastic integration with a constant Jacobian, ensuring control dynamics in our simulation. In our approach to neural mass modeling integration, we use the Wang trick with exponential matrices for efficiency. And I want to highlight a basic observation that simplifies neural mass models by decoupling them with a sufficiently small integration step. The input to each neural mass can be assumed to be available. In turn, this means that from the point of view of any given neural mass, its inputs are already available. This innovative approach represents a significant step forward in addressing the challenges of neural mass modeling. In this slide, I want to briefly show you how was the evolution of the local linearization method in neural mass modeling. Firstly, it was applied by Valdez-Souza for parameter estimation to autonomous stochastic differential equation. It did not incorporate transmission delay, which were not generally modeled at that time. After, we have Sotero and once again Valdez-Souza, and they work with non-autonomous random differential equation. They also include the Wang trick for accelerating the numerical integration, and they incorporate Dirac delta function to interpret transmission delays. After that, we have a group of researchers that work with autonomous stochastic differential equation, and they interpret transmission delays through a Dirac delta function. And an example of that is the stochastic DCM. Later, we have published a paper where we work with non-autonomous random differential equation. We also keep working with the Wang trick, since it's a very good strategy to increase the speed of the calculation. And in this time, we include the distributed delays to analyze the transmission delays in signal propagation. As previously mentioned, I followed the non-autonomous scheme of the log linearization method for random differential equations. The numerical integration methodology is based on the following steps. The input for each neural mass is a combination of time shift versions of other neural masses outputs and the connectivity strengths, and this information is included into the connectome tensor. The system of equation is solved with a sufficiently small integration step, making the input to each neural mass known. Thus, the solution of the random differential equation can be carried out for each mass in isolation. This approach uncouples the integration of each neural mass with significant simplifications in the solution of the system of differential equation, which are now low-dimensional. The integration for each neural mass can be solved symbolically and explicitly expressed. For that, I use the MATLAB symbolic toolbox. Now the toolbox and methods were tested with an extended neural mass network for a much larger number of cortical columns, in this case 1,000 cortical columns, which means 3,000 interconnected neural masses. Since our goal is to describe the whole brain, in this case each brain region could be modeled with a single Janssen and Reed unit, and this is a very high dimensional problem, and the developed toolbox don't spend that much time in solving the model. We turn now into examples of the use of the model and our toolbox, and these examples are only to illustrate some use cases of our method. This figure shows changes to the Janssen and Reed output spectrum when the input parameters to the stellate cell are modified. As seen, oscillatory activity is reflected in spectral peaks that depend critically on these parameters. When the mean of the noise input is above a threshold of 25, regardless of the standard deviation, a clear alpha peak appears, and harmonics as well. However, when the standard deviation increased from 0 to 1, the number of harmonics decreased from 3 to 2. And on the other hand, for a mean below the threshold, the primary peak shifts to the theta range, and the number of harmonics increased to 7. We investigate the spectral dynamic of the system under different delays scenarios, specifically Dirac delta delays and distributed delays. Additionally, we consider 3 distinct connectivity topology, and we observed that the generation of alpha rhythm was achieved for Dirac delta delays. However, when we introduce a distributed delay, the oscillatory activity shifts from alpha rhythm to theta waves. Since the neural mass model has a higher variability, we decided to make a sensitivity analysis of the larger formulation of the Janssen and Reyes neural mass model. And for that, we make a parameters space. And to do this, the neural mass model is simulated a large number of times using different parameters, which are chosen using a Latin hypercube design. This is a space filling design which allows to efficiently explore the whole parameters space given a fixed number of simulations. After that, we make a simulation classification where each simulation is then classified in terms of some chosen characteristics. And here we chose to focus on characteristics that are often used to define healthy and epileptiform rhythm. An example of that are the amplitude, the frequency, and the number of peaks per period. The final step is to feed the data with a statistical model, and here we choose to use a tree approach, which cuts the parameters space into rectangular regions of different sizes and is amenable to high-dimensional analysis. The sensitivity analysis was made for 2 types of dynamics, the steady state and seizure neural dynamics. The steady state dynamics is a stable and consistent patterns of neural activities with minor fluctuations indicative of normal brain function. And the seizure dynamics characterized by high amplitude and high frequency oscillations often associated with abnormal brain activities such as epilepsy. So taking this in account, we make our sensitivity analysis and analyze the higher importance parameters. In this slide, I am showing the results of the sensitivity analysis for the steady state and the seizure dynamics study. And these scores indicate the contribution of each predictor to the overall predictive performance of the model. And as we can see, I also include the realistic parameters for time delays analyzed, like all the axonal properties involved in this computation. And as we can see in this graph, in the case of the steady state analysis, the slope of the sigmoid function show higher importance, and for the seizure dynamics, the maximum amplitude of the postsynaptic potential for the excitatory cells is the parameter with the greatest impact on the model output. It is important to notice that for the axonal properties, they also play a role, an important. Making a summary, this work covered general formulation of neural mass model with a focus on the neural connectome tensor concept, and efficient algorithms of the distributed delay connectome tensor neural mass model was presented for integrating high-dimensional neural mass model. Distributed delays were realistically calculated using anatomical data for example, the axonal properties like the length, the diameter, the myelination, the G ratio. Emphasis was placed on the significant impact that distributed delays between neural masses can have on dynamics. Additionally, it was explored the extension of the Janssen and Reed model, specifically in the context of 1,000 cortical columns, and explore how various connections topology influence brain dynamics at a large network level. And lastly, sensitivity analysis showed the most important parameters for steady state and seizure dynamics. So right now we are working on the incorporation of experimental data as input to the connectome tensor. We are working with the human connectome and big brain dataset to give a better description of each layer. So we are also looking for ways to incorporate neurotransmitter receptors and intracranial EEG recordings to the model and link this neuromass model to the fMRI model developed by Camille. So that's all from my side. Thank you so much for your attention, and I hope you really enjoyed this talk.